All right, let's get started. Welcome to microbiology. I'm Professor Leonard. Hope you're all staying warm. One of my colleagues, it was so cold in his office that he took his laptop into the walk-in incubator and was doing his work in the walk-in incubator. So, it is cold out there. I often will start microbiology lecture talking about why you should be interested in microbiology. Go ahead and just come wherever you can fill in. There is 130 of us, and this room holds about 140. So we're gonna be packed in, especially on exam days. So we get used to it. I often start class with why you should be interested in microbiology, but you know, we're in a pandemic, right? So that speaks for itself. I hope that what you learn from this class will help you make good decisions, you know, for yourself and people you love about, you know, how to conduct yourself in a pandemic. So this class has a very clinical bent to it. I think you'll see that as I talk about, you know, what we're going to talk about this semester. Microbiology is so much more than that. Like we could study microbes in the soil that would help, you know, get rid of pollutants. And that's a really cool aspect of microbiology. But we kind of focus on how microbes are bad and how we can get rid of them. So again, there are lots of good things that microbes do, and we'll touch on that as well. But, all right, so we're gonna talk through the syllabus just a little bit. I do hope that you will take the time to read through the syllabus. I'm just gonna hit the highlights. And it looks like there is a back door to this room. So if you know that you're going to come in a little bit late, you'll have to try to figure out how to come in the back way in the future. It's kind of a bad setup for this room. All right, so my contact information there is in the syllabus. My office is right down the hall from the labs. So I know that probably most of you are in micro lab right now, and I do kind of talk about lab and tie that into lecture quite a bit. If you're not in lab, don't worry. I'm not going to test you over anything in lecture that you have done in lab, unless we talked about it in lecture. But I do refer to it quite a bit. So this is my office hours right now. If you need to see me other times, those times don't work for you, don't be afraid to talk to me, get in touch with me. I'm mostly here on Fridays. Just, you know, touch base with me and make sure that I am, and I'd be glad to meet with you on Friday because I don't have any classes Friday. I usually come in for the seminar if we're having a seminar at noon. I do teach 3 labs. So, you know, the rest of my schedule is kind of filled in with teaching labs. And partway through this semester, I'm actually a track coach for a high school that's close by. So that those hours of like 3 to 5, at least partway through the semester, are not going to be good hours to track me down. So more available in the mornings, I guess. A little bit about me. So this is my family. Took this picture during not this summer, the summer before. Some of you know my husband. He is Dr. Leonard. He's in the chemistry department. You can call me Dr. Leonard and I answer to that, but I actually don't have my PhD. I just have a master's, but some of you may have run into him. My son there, he's our youngest. He's got his pandemic haircut there. Now he's got a hire me haircut. He's in his 4th year at Michigan. Incidentally, just came home because his roommates have COVID. So it was blowing up on that side of the state. And obviously we're worried that it will here too. Or really it is already. Right? What else? Our middle daughter, she has autism. She lives at home with us, and she just got her first part-time job, so we're really excited about that. And my oldest daughter, she's in her 2nd year of medical school, just started clinical rotations, and her first one is in Hawaii. So she's in medical school through the Navy. And somebody's got to do it in Hawaii, right? So she just flew there yesterday, so we're kind of excited about that. But if you're interested in, going to professional school through the Army Navy, then talk to me about that. It's really a great way to go. They pay for pretty much everything, so it's pretty. Talk to me more about that if you're interested. I have been teaching this class, I think, enough that I'm kind of losing track, like 7 or 8 years. And I've taught lab for about 15 years. So I like this class. I'm really interested in it, and hopefully you will be too. So textbook, let's get that out of the way. So this is my first semester with the SAVE program. I think maybe some of you did that last semester, but this is new to me. So students who are telling me I'm having trouble accessing this textbook through the SAVE program, that was because I didn't have it set up correctly. So about an hour ago, I got this, I think, mostly figured out. Now there's the very first tab on Blackboard is, SAVE or whatever. And it should, hopefully, a number of you have done that successfully, maybe 20 of you in the last hour or so. So click on that, SAVE, and it should walk you through it. If you already purchased the textbook, that's okay. I actually have, now it's going to be up in like 2 different places, what I see, but it should be fine for you. If you purchased it yourself though, you will have to opt out of the SAVE program because they're automatically billing your tuition bill for the textbook for this class. So if you only got temporary access and you didn't pay for it yet, you might want to go through the SAVE program if and when you go to purchase it. Okay, and that I really like the textbook. I really like the whole online component of it, Mastering Microbiology. Students, I get good feedback that they like the homework, and obviously that had to grade a little bit. We'll talk about that too. So I don't know, through SAVE program, does anybody know what they're charging you through that? Is it like 100 or was it less than that? 90. Okay, so they're saving you set $10 by going through the bookstore because I think it's $99 if you purchase it on your own. So I will try to answer any questions you have about that. If you have questions about opting out, I think you might have to contact the bookstore. Send me your questions and I'll tell you whether I can help you or whether you'll have to just contact Pearson. But inevitably there will be a little bit of miscues. So if you go through the SAVE program, you won't even need that code. This is if you do it not through the SAVE program. Sorry about any confusion there. I'm learning as we go too. Okay. All right. Grading. So I just got through reading the evaluations from last semester, and one of the complaints that students have is I don't like the way this class is graded. It's mostly exams. And if you look at it, there is only 10% of your grade here that comes from the homework. And it is mostly exams. That's just the way we teach this class. We can't do discussions. We can't do group projects or anything like that very easily. So it kind of is what it is. I did, after last semester, I think it might have been just an artifact of students going through classes during the pandemic and having online stuff. I found that when I gave the first exam last semester, a number of people just really flopped it. They really did poorly. So I decided this semester that I was going to make that first exam work a little bit less. So the first exam is going to be a little bit shorter. It's going to cover less material, and we're going to do it earlier. You know how you always got, you get to a certain week in the semester and you've got like 5 exams in 2 or 3 days. All right, I'm going to do our first exam the week before. So we're only going to have 4 lectures, and then we're going to have an exam. I'm just going to try that. Let's see how it goes. We'll talk more about what that exam will look like, but they're all multiple choice. I do have and I like to have the final exam work a little bit more because I always have students that kind of do better and better and better, and they can get the hang of things, how to study as the semester goes on. So like to give them a chance to maybe make up for an exam, the early exam that wasn't so great. So it does put a little bit more pressure on the final, but you can kind of look at it two different ways. Other professors, so the four of us that are teaching this course right now, we all talk to each other quite a bit, and we try to kind of match up what we're doing. Some of the other instructors have this worth a little bit, maybe different amount of points. You might see a little variation there in terms of how much the homework is worth, for instance, but we do all try to have our final averages about the same. So one of the other professors, for example, he makes his homework worth more points, but he doesn't curve his exams as generously as what I do. So there's going to be some differences there. The homework, I've written extensively about the homework and how that's graded in your syllabus. So I'll refer you to that. But you should all earn all 60 of those homework points. Because the way I have this set up is that if you get 80% of all the homework points, then I'll give you all of them. Okay, so for example, if there's 500 possible homework points, 516, and you earn 413, then you get all 60 points. The reason that I did that is because that I found that students were spending hours and hours on the homework trying to get every little point right. And that's not really the point of it. The homework is just is meant to be practice, review for you. You know, I want you to do it carefully enough that maybe you're looking up some things and really thinking about it, but not so careful that it's just taking you forever. Okay, I think that to some point that's detrimental. So most of the homework points are review exactly. I've chosen questions exactly what we've talked about in class. Some of the questions might be more geared towards getting you interested, like there's some case studies, that sort of thing. Occasionally, there'll be like a 5-part homework question, and 2 of those parts maybe we didn't talk about in class, but the other 3 parts are so good and exactly what I want to ask you that it's kind of worth throwing them all in. So if there are any questions about a homework question, like you think it's not fair or it's incorrect, what they say is the right answer, don't be afraid to shoot me an email and we can talk about that. So again, most people like doing the homework, and it gives a little bit more practice. Okay. So how to do well in this class? Like that's a question that you should be thinking about. Hopefully you haven't heard too many horror stories. We do cover a lot of content in this class. It's not per se hard to understand, but there is a lot of it. And it's going to take a little bit of kind of chewing on to kind of shift things to your long-term memory. It's always better to do that than to try to memorize things a day or two before the exam, because then you'll forget it when it comes time for the final. You'll see that I posted PowerPoints. I try to post them at least the night before. My goal is to have them all posted at the beginning of the week for the whole week. So I post the outline, the PowerPoints, and then I will add information to that as we lecture. So I do that with a tablet and I write in that information. You do what works best for you. I have a number of students that are using tablets and doing the same thing that I am. You could be typing it in on your laptop. You could be printing those PowerPoints out. I would suggest maybe 2 to a page rather than 3, because sometimes we get, it gets pretty small writing. But, let me know what works for you. And if you're having trouble figuring out what works, then I could help you with that too. But you will need to take some notes in class. I do post learning objectives, and actually I've got those posted for the whole semester already. So this is, maybe a 10-page document. I like to print it out because when I'm making up the exams, I have all my exam questions filed under kind of what objective they're covered. So I go through and I grab a question. Oh yeah, that fits under this one, and this fits under here. So some students go through and they write out the answers to all these objectives, and that's a ton. That's a lot. I would probably use it more as, a few days before the exam, kind of check in oh, what's that? I don't know what that is. Go back to your notes and look it up. But again, it's a guide. It's one tool that helps people. I try to make all my exam questions come from these objectives. Every once in a while, there I might throw in something that doesn't fit exactly, but that's what I try to do. All right. So and I test you over what we cover in class, not over the readings. Okay. So the textbook is meant to just kind of complement. I mean, it's never going to hurt you to read the textbook. I definitely would encourage you to do that, but I would use it probably more as, oh, here we talked about viral replication. This was really confusing, how she said it in class. I'm going to go back to textbook and read about it. And then maybe that's going to make sense a little bit better. So it's just kind of another way to see something again to kind of help it sink in. If you, of course, when you buy the textbook, that's an e-text. And if you want to have a hard copy of the textbook, you know, you can purchase those. We're using the 6th edition. The 5th edition is almost the same. So if you really like having you know, a real textbook, I would buy the older edition. It'd be a little bit cheaper, or even the 4th edition is pretty close too. So the homework, I have all the homework due the same day as the exam that covers it. So, but I would encourage you to kind of do the homework as we go. So after we get through chapter 1, you know, I might remind you and say, hey, we're done with chapter 1. So, you know, next day or two, go ahead and start working on chapter 1. Okay. Terminology is big. So in any biology class, you should have noticed this, like, you know, and it kind of builds on each other too. So, you know, once we define the word enzyme, I'm going to use that like you know what it is. So you want to make sure, okay, you know, oh, an enzyme, oh, it's a protein. Oh, it's made up, that means it's made up of amino acids. And so you got to make sure you're learning that terminology. I have bold typed some of the terms that I'm pretty sure are going to be new for you in the learning objectives. So that's one tool. Hey, do I know what that word is? And it's part of the final will just be all terminology. So it'll be matching of terms. So again, just encourage you to learn that terminology. And I talked about the textbook a little bit as well. Part of the very end of the syllabus, I know it's kind of a long syllabus, but at the very end, there's a whole section. I read a book last summer about basically how to study. It was called Learn Like a Pro, and had some really good tips. If you're a procrastinator, I passed one of those tips on to help you not procrastinate. Just some active studying tips. So as much as you can, when you're studying, don't just read over your notes, but cover part of it up. Oh, what is the difference between an electron microscope and a confocal microscope? I have no idea. Okay, look back again. Okay, now quiz yourself again. Can you get some content in there? All right, so as much as you can kind of practice. Sometimes it helps to do that with another person. Sometimes that might be distracting. Sometimes you want to teach a stuffed animal or teach yourself in the mirror or something. And that will test if you really know something or not. Okay, so whenever possible, be as active as you can. Okay. So what are we going to be talking about this semester? The first really couple weeks, 2-3 weeks, are all just kind of basic stuff about microbes. And we're going to talk a little bit about what microbes are today to start out with. We skip around a little bit to begin with, and I didn't post this just because it's there in the syllabus, much better, written a little bit neater. We skip around a lot to begin with here because we're trying to match up a little bit with lab. All right, so you're talking about aseptic technique in lab. So we're going to skip and talk a little bit about growing up organisms in the lab a little bit to begin with. And then we're going to go talk about microscopy because you're going to be doing that in lab. So again, it seems like we're skipping around a little bit here. We'll kind of settle down a little bit past these first few weeks. Cell structures, enormously important as we move forward. We are going to kind of build on that quite a bit. And again, that first exam, that's really just in a couple weeks. That'll be here before you know it. Talking about how to, what microbes need to grow, and then how they grow. So what kind of metabolism are they using? Don't be afraid of metabolism. We're not going to be memorizing pathways like maybe you have in biochem. We're going to do more of an overview approach, but bacteria can do some special things that we can't. So we're going to focus on those things that they can do. A whole section on genetics, then how they reproduce themselves. So some of that material is going to be familiar depending on other courses that you may have had. And then we get into, after we talk about viruses a little bit, we get into a very practical part of the course. How do we kill microbes if that's our goal? So what kind of disinfectants we can use, or physical methods to kill microbes. What a little bit about antibiotics. How do we kill, how they work, how in our bodies, and then kind of a process of infection, and how does our immune system work to fight against microbes. So again, very, very clinical focus, and whatever time we have left, we talk about some particular infections like tuberculosis, like anthrax, and we kind of pull in stuff that we've learned all semester specifically about those infections. So that's a little bit of an overview of the semester, what you can expect. Any questions so far? Homework, exams, anything like that? Okay, again, refer to the syllabus there and ask as you think of those questions. Before we move forward into chapter 1, I just wanted to make a little plug for the test prep course. So if you've never been to the class advising center and you're planning on going to some professional school, like you want to go to dental school or you want to go to medical school, you should make an appointment this week to the class advising center, because they're really helpful over there. They can tell you what kind of experiences would be helpful for you to be getting, what kind of jobs you should get. If you're planning on taking the pre-dental exam or the MCAT this spring, here's a great way to review for that. So you can take a Kaplan course, which will cost you what, $3,000. Like, those courses are awesome, but if that's not in your budget, you're not planning on doing that, something like that. This in the past has been about $60. So you go once a week, or maybe even it's virtual. I'm not really sure, but this is a great way to review, to get some help to review for some of those exams that are so important. So encourage you to take advantage of that. Okay, so chapter 1. Chapter 1, I always like to tell you a little bit about what we're going to talk about before we talk about it. We're going to start out with, what do microbiologists study? Okay, they study microbes, things that are too small to be seen with the naked eye. Little hint there. Jump right into what causes fermentation. That's kind of a confusing thing, I think, for a lot of students. What is fermentation? But we use it in the sense of that's what bacteria do, and other organisms like fungi, like yeast, when they're changing sugars into end products, either acidic end products or alcoholic end products. So we're going to talk about some historical figures that's one of the early problems that microbiologists worked on, understanding what causes fermentation. So we're going to talk about the contributions of Louis Pasteur, and then several other people, including some people that did work about what causes disease. We still have to think about that today. Certainly not something that we only thought about in the past. And of course, what can we do to prevent disease, and people that have made contributions in hygiene and vaccines, things like that. So chapter 1 is mostly a little bit of history. So if we think about what microbiologists study, well, we had to be able, really helpful to be able to see things that are smaller than what our naked eye can discover. So who made the first microscope? Usually that person that's given credit for that is Antony van Leeuwenhoek. All right, and the years are not important to memorize here, but just to give you an idea, you know, he did his work mid to late 1600s, early 1700s. So we're talking quite a while ago. He was a cloth merchant, Leeuwenhoek. And that seems to me like he was a cloth merchant and he made a microscope. Like, how do those things gel together? Well, think about it. He was Dutch, and obviously there's a lot of Dutch people that live in this area. And when we moved here, we learned all the stereotypes that we have for Dutch people. Like, okay, they're tall, right? They're really neat and tidy. They clean their houses all the time. Another stereotype that exists about Dutch people is that they are thrifty. We'll say it that way. I don't want to say cheap, but they're thrifty. Well, that described him. He was a cloth merchant, and he wanted to make sure that if he was paying a lot of money for some fine cloth, he wanted to make sure that it actually was good quality. So he got really good at crafting his own lenses, and so good that he could actually have as good of magnification as what we have in our scopes in the lab, somewhere between medium and high power. So he was actually getting pretty good magnification with something that looked like this. That was his first microscope. So the lens is actually right here. And then there's kind of a little pointer right there, and he could stick his specimen on there if he was looking at a fly or a piece of cloth or whatever. And then I think he would kind of hold it up and look under it like that. So it must have been kind of tricky getting the light just right. But again, he looked at all sorts of things. Let me write this here. First microscope. He looked at all sorts of things, you know, pond water, you know, cloth, you know, his own bodily fluids. And he wrote up, you know, what he saw in scientific papers and submitted them to the Royal Society of London. And he was pretty well known in his day as, you know, being a respected scientist. The problem is he never would tell anyone how he crafted these lenses. So making microscopes didn't really advance. It took a long time because he didn't share how he was doing that. Okay. So he described what he saw under the microscope, the living things he described as wee animalcules. So if we were to think about these wee animalcules that he saw, we could think, oh, what kind of things was he seeing? He was seeing things like bacteria, archaea. We'll talk about the difference between those two things shortly. Probably some fungi, like yeast are unicellular, protozoa. Let's see, what else? Algae, I always forget algae. And maybe some small multicellular animals. It's an M. Okay, so those are the kind of things that he saw. The other thing that we would consider a microbe, for sure, I'll put it in parentheses. I'll put viruses in parentheses because obviously he could not see viruses. We weren't able to visualize viruses until the 1930s when we came out with electron microscopy. Okay, so we knew that they probably existed before that, but certainly not that long ago. And then another name that we want to remember, Carolus Linnaeus. He was typically called a Swede. He was from Sweden. He would probably call himself a botanist. So his life overlapped with Leeuwenhoek's quite a bit. He was a little bit younger. He is best known today, we would consider him the father of taxonomy. Right, so we credit him with really helping to organize the scientific community about naming organisms so that we could, know that we're communicating about the same organism. All right, so we're going to talk more next lecture about taxonomy and why that's important. But he came up with, for example, using Latin names to describe organisms and the whole kind of genus, species, taxonomy scheme. We'll talk more about that. Okay, so as Leeuwenhoek was describing all these animals that are, that we never knew was there before, it became important to be able to classify them in some way. And we've been refining taxonomy ever since. All right, so what are some of these things, and some of these classifications we'll talk a little bit more about later, but this kind of is an introduction. Prokaryotic organisms, the bacteria and the archaea, those are both considered prokaryotes, and that just means they don't have a membrane-bound nucleus. All right, they still have DNA, and it's all in kind of one spot in the cell, but it doesn't have a nucleus around it. Okay, so I just threw up some examples here of pictures of bacteria. This is a what these are. Don't write that down. That's not something to memorize. This is called a Deinococcus, the one on the left there. That's an organism that can actually grow in radioactive waste. Right, so a lot of bacteria are really helpful in helping us, break down pollution or toxins. This one is Staph aureus, a common bacteria that causes skin infections and lots of other stuff. You'll work with that organism in lab a little bit. Both of those are bacteria. This one here on the right is considered in that archaea taxa. That's an organism, it's called a methanogen. It actually produces methane. Okay, so what's the difference between bacteria and archaea? One difference is, well, there's a lot of kind of slight differences. One difference is the cell wall structure. There's a particular compound that's unique to the bacteria that we don't see in any other life that has a cell wall, whether it's archaea or whether it's something like a plant or fungi. So we'll talk more about that later. The archaea are often found in kind of extreme environments. Although we know that there are archaea that are actually part of our normal microbiome. But when we kind of think of them, we think of, oh yeah, those are the ones that live in the Great Salt Lake or on the polar ice caps or on the deep sea vents. Okay, they have some kind of strange metabolisms, you could say, that enable them to do that. And as far as we know, none of the archaea cause disease. So who knows in the future that, there's an exception to every rule, it seems like, but as far as we know, we haven't found any yet that are harmful to us. Okay, so those are the prokaryotes. And in general, I would say, I don't know how to write this, both the bacteria, so both are smaller than eukaryotes. Okay, usually a prokaryotic cell might be as much as or more than 10 times smaller than a eukaryotic cell. Okay. Some of the eukaryotes that we're going to study. We're going to spend a little time on fungi, particularly fungi that are microscopic. Okay, so over here we have yeast. Okay, most college students know the use of yeast as doing alcoholic fermentations. Here's another filamentous fungi. This is Penicillium chrysogenum. It secretes penicillin. So that's been a really important fungi in terms of our human health. We'll briefly talk about protozoa. Here's some examples of protozoa. They are ubiquitous. That means they are everywhere, especially in bodies of water, things like amoeba, paramecium. There are some protozoa that are going to cause us harm. We'll spend a little time talking about malaria later on. And then algae, we don't talk a lot about algae. We'll talk about how they're classified basically, and obviously really important for food chains and producing a lot of the world's oxygen. But they, for the most part, don't cause us any harm. They don't cause disease, except a certain kind when there gets to be way too much of it. But we don't spend a whole lot of time on that. And I will briefly mention parasitic worms, just because when we think about disease, sometimes we think about things like roundworm or tapeworm or hookworm. There are some parasitic worms that might have a microscopic part of their life cycle, like their eggs might be microscopic, but we're not really going to talk about them anymore in this class. We do have a parasitology class here at Grand Valley. Dr. Graham teaches it, and I've been meaning to sit down on it sometime myself, because he's had a lot of the parasitic infections that he teaches about. So it sounds like a very interesting class. And lastly, we're going to spend some time on talking about viruses. Okay, so incidentally, this is the graphic that I've used for many years when I introduce viruses. That is the SARS coronavirus. So SARS-CoV-1 causes severe acute respiratory syndrome. The graphic that I've just had up there for a long time. Basically, what is a virus? It is some nucleic acid surrounded by a protein coat. Okay, and then sometimes there's a membrane around that, but sometimes not. So we're going to learn more about that. We consider them acellular. So that means they don't have a true cellular structure. They don't have a nucleus. They don't have ribosomes. They don't have organelles. All right. They can only reproduce when they're inside a living cell. Okay, so do we consider them living organisms? We'll talk more about that later on. Most often, scientists would come down on the no side of that. But of course, there's viruses that infect animal cells. I consider humans an animal. There are viruses that infect bacteria cells. And some of them have this really bizarre shape. They look like little robots almost. We call those bacteriophage. So we'll talk about those as well. All right, so moving on to some problems that early microbiologists encountered. And sometimes it's really interesting, and I think good for us to think through some experiments, early experiments, because it kind of reminds us how science works. And your book uses the example of how Louis Pasteur applied the scientific method to solving the problem of how fermentation works. So I know you all know what the scientific method is, but again, it's important to kind of remember how we only want to have one variable at a time, right? We can't run clinical trials and change a whole bunch of different variables, because then we wouldn't know, what's the best dose, or whether, the vaccine works or not. So we try to have it very carefully controlled. So that's good experimental design to have good controls. So what we knew about fermentation at the time. So this was the time that Louis Pasteur was doing this was 1861. So Civil War period in our country. Louis Pasteur was French. And of course, a really important industry in France back then, and still is the wine industry. And we knew that we could take grape juice and it would ferment into alcohol, but at that time we didn't have any idea how that process worked. And when something would go wrong, then we didn't know what to do to fix it. So vintners were having problems with their wine spoiling. So they contracted Louis Pasteur, who was a trained chemist, to try to help them figure out what was going on. So they knew that when grape juice fermented into wine, they could look at that wine and they knew that there were yeast and bacteria in there. So Louis Pasteur set up an experiment. So his first hypothesis, well, maybe this just happens on its own. Okay, spontaneous fermentation occurs. So in order to test that, he took a flask of grape juice, heated it up enough to kill any microbes that might be in there, and then sealed the flask. Waited a certain amount of time, and then looked at the contents of that flask. Well, there weren't any microbes, and there was no fermentation. It was still grape juice. Okay, that's not the right hypothesis. Maybe it's just the air that ferments grape juice. Pasteur had designed these what he called swan-necked flasks. If you can read about this in your book, I've decided to skip over it, but the whole problem of where does life come from? Does it spontaneously generate? He helped solve that problem with designing these flasks, actually. But what was special about these flasks is that they would allow air contact with the grape juice here, but anything that was floating around in the air would kind of settle out in that tubing. Okay, so he boiled the grape juice once again. Air had access to it, but still nothing happened. No fermentation, no microbes. Well, maybe it's the bacteria that ferment grape juice into alcohol. So once again, he made sure that the grape juice was sterile. He added bacteria, sealed it up. And what did he find? Lots of bacteria. And something that we know now, and you will test this over and over again if you're in lab this semester, is that oftentimes when bacteria do fermentations, it's almost always acidic end products. So things like lactic acid, acetic acid. Acetic acid is vinegar. So when you are expecting to drink wine and it tastes like vinegar, right, that's not what the vintners wanted. So, so bacteria do carry out a fermentation, but not the kind that we want. And lastly, do yeast ferment the grape into alcohol? Again, he sterilized that grape juice, added the yeast, sealed it up, and got lots more yeast and the alcohol that they wanted. So again, examples of using a scientific method to solve a problem. So that brought about this process that he called pasteurization. So the vintners would heat up the grape juice and then add the yeast, and then to get a reproducible wine product. We don't actually pasteurize wine anymore. We do that chemically because heating the grape juice up changes the flavor of it a little bit. But we pasteurize lots of other things, and we'll talk about that, how we do that a little bit later in the semester. Pasteurized milk, for example. A contemporary of Louis Pasteur, he was slightly younger, was Robert Koch. Okay, and Robert Koch was German, and he was interested in solving a lot of the same problems that Louis Pasteur was. And together they really are credited with ushering us into this, what's considered the golden age of microbiology. So from about 1850 to 1910, we made a lot of advances in microbiology. And again, these two men have had a lot to do with that. So they were working on the same things, but they're really competitors. They didn't like each other very much. So unfortunately, instead of collaborating, they were kind of always calling each other names and writing letters and things like that, unfortunately. But Robert Koch was most interested in what causes disease. And we refer to that as disease etiology. Make sure I spell that right. Okay, so that means the causation of disease. So, up until, we started to understand really 1850s, 1860s that it was usually microbes often that were causing disease. Before that, there was, all sorts of theories. Most people thought that it was bad air that caused disease. So malaria, bad air, was just thought to be, you're close to a swamp or something like that. So with the germ theory of disease, we were starting to understand, very often it's some kind of microbe. And Robert Koch was the first to prove that a microbe caused disease. All right, he discovered that it was bacillus, a bacteria, Bacillus anthrax, he called it, Bacillus anthracis, that caused anthrax. And he was known for very meticulous experimental design. For most of his life. He not only discovered that a bacterium causes anthrax, he also discovered the causative agent of tuberculosis, which is a huge scourge at that time. Later in his life, he thought he also had a cure to tuberculosis, and he had kind of gone off the rails a little bit later in his life and kind of said some things that weren't true. But again, for most of his life, he was known for this meticulous experimental design, and he came up with a set of rules that are known now as Koch's postulates. And these are just a series of things that need to be satisfied in order to prove that something causes a disease. All right, so let me illustrate that. There is not a graphic in your textbook that talks about this, although they do talk about it in your textbook. But what are Koch's postulates? So these are things that kind of seem obvious to us now, but back then, not so much so. So basically, if you're trying to prove that a particular agent causes a disease, you had to go through these series of experiments. First of all, the germ should be present in every case of the disease. So if you had mice, say that you infected on purpose with the infection, you should be able to take a tissue sample from that mice and see that particular bacterium, hopefully through the microscope, as well as plate it out on some media and grow it up. Okay, and isolate it all by itself. So only that one germ was growing in that media. And usually that germ is not going to be found in a mouse, say, that was healthy. All right, so again, this is some other germ, not the same one. I mean, okay, so that wasn't necessarily easy, because the way that we were able to visualize microbes under the microscope at that time was not very advanced. Our culturing methods were not very advanced, so that wasn't necessarily easy. And then you had to take that given microbe, infect a healthy animal, and be able to do the same thing again. Be able to, that animal would get sick, you could see that microbe under the microscope and grow it up on some media. So if you could do that, then that was pretty good evidence that that microbe caused that particular disease. Okay, so that was really helpful. Of course, we know not all diseases are infectious. Sometimes we can be healthy carriers of a disease. We can be carriers of whooping cough, for example, and not actually have whooping cough. So there's some diseases like pneumonia that can be caused by lots of different things. So there are things that kind of muddy the waters, but this was really a helpful first step. And then Hans Graham, he was a worker in Koch's lab, and he developed the gram stain in 1884. You're going to be doing lots of gram stains if you're in lab this semester. But this is a really helpful staining procedure because it has helped us understand that there are really two different kinds of bacteria, ones with a certain kind of cell wall, gram positives, and a different kind of cell wall that we call gram negatives. So when you do this special staining procedure, the gram positives stain one color and the gram negatives another. And that's generally the first question we ask when we're trying to identify an organism: is it gram positive or gram negative? So we will learn much more about that. In terms of preventing infection and disease, now that we understood it's microbes that cause it, we made a lot of improvements in basic hygiene. Okay, so how do we prevent infections in a hospital, for example? We got to wash hands. We got to make sure that they're clean conditions. So some people that we remember from making advances here, Ignaz Semmelweis was a Hungarian physician. So this is 1848. Again, you didn't necessarily need to memorize dates, but this was kind of before Pasteur had done his work, before we really understood that germs can cause disease. And he advocated hand washing. So he noticed in his hospital that surgeons would go from doing autopsies on dead bodies and go straight to maternity wards and deliver babies and not even wash their hands. And the death rate for childbed fever for women when they were giving birth was pretty high. It was like 20% in his hospital. So he started having his surgeons wash their hands before they deliver babies, and that infection rate and the death rate went way down. Unfortunately, other doctors kind of, took insults that they were the ones causing women to die, and they didn't really understand why hand washing helped. So he eventually lost his job and ended up dying in an insane asylum. So that was not a happy ending for him, but he did write that up. And, a number of years later, John Lister, he was an English doctor, and he knew about Semmelweis's work, and he advocated doctors washing their hands in carbolic acid. And another name for carbolic acid is phenol. Phenol used to be the real kind of go-to disinfectant. It's kind of a little bit more toxic, caustic than what we like to use now. But this was really, in fact, it worked really good. So doctors would wash their hands before surgeries, much lower infection rate. He would spray carbolic acid in surgical operating rooms. The downside was that it gave the doctors respiratory problems from breathing in this disinfectant. It turned their hair, bleached their hair white. They lost their fingernails. So there were some side effects there, and refinements needed to be made in that, but it was a step forward. Florence Nightingale, she was a nurse in the Crimean War, which was from 1854 to 1856. And she just advocated high standards of hygiene. All right, so on the battlefield, keep those soldiers clean, keep their wounds washed out, keep the hospital clean. And she kept really good records and statistical analysis so that people believed her when she said, hey, keeping things clean saves lives. All right, so 3 people we remember for really helping to improve our hospital acquired infections. If we're talking about history of microbiology, I gotta mention Jon Snow. He's thought of as a father of epidemiology. Sorry if my handwriting is not great there. I should say epidemiology. So epidemiology is kind of the study of how diseases are transmitted. It's a lot of asking questions. It's a lot of statistical analysis. At the time, 1854, there was a big cholera outbreak in London. And at that time, we didn't understand what caused cholera. Now we know it's a little curved bacterium called Vibrio cholerae. But that's a bacterium that lives in contaminated water, right? People drank the same water that they defecated in. Jon Snow went around to every household and he asked each household questions. What have you been eating? Do you have anyone sick with cholera? Where do you get your water? All kinds of questions that he could think of. And he, made a red dot here on this map for every household that had somebody sick with cholera. And he realized that they were centered around this blue dot, which was the Broad Street pump. So most of those households went and got their water at that pump. So he took the handle off the pump so nobody could get water there anymore. And the kind of epidemic there subsided. So it's just kind of the first one to just apply some methods to try to cut down on an outbreak. And in terms further of preventing by vaccines infections and actually treating an infection when someone has it, a couple of people we remember, Edward Jenner. Okay, so again, this is early 1796. He's credited with the first vaccine, a vaccine for smallpox. He noticed that milkmaids that contracted cowpox hardly ever got sick with smallpox. And so he tried an experiment. He took some of the sores from a cowpox infection on a milkmaid and rubbed them into the skin, not rubbed it, but cut that, that scab into the skin of a boy. So he got cowpox. And then a few weeks later, he actually inoculated the boy with some smallpox lesions. So not the way we do clinical trials today, but the boy didn't get smallpox. So it became a very common practice. It had its disadvantages and risks associated with it, but smallpox has usually like a 30% mortality. So in terms of risk assessment, it was probably a good idea. Paul Ehrlich, he was another German scientist that I believe was in Robert Koch's lab. We think about him as being the first one to try to develop a drug to treat infection. So he came up with a treatment for syphilis. He was known for coining the term magic bullet. So I'll write that here because sometimes you hear that. So if something is a magic bullet, it's going to kill your enemy and not harm your friend. All right, so he was searching for a drug that you could take and it would kill the microbe that was causing the disease and not harm our bodies. Okay, that's something that we still have a huge problem with today. We have a lot of antibiotics in the pipeline, but a lot of them are too toxic for us to take. So again, they're not worthwhile if they're too toxic for us. So he searched a number of compounds trying to find one that was effective against syphilis. He finally found one that had arsenic in it that had some use. Unfortunately, arsenic is pretty toxic for us, so not widespread use, but it was a first step. Okay, so this is Paul Ehrlich down there. Okay, so that's pretty much the history that I want to impart with you. Chapter 1, if you read it, there's some other names mixed in there. If you're interested, by all means read that. But you could be working on chapter 1 now because that's pretty much what I'm interested in having you know on chapter 1. And again, I try to pick homework questions that ask you about that material. Okay, don't be afraid to raise your hand, jump in if you have questions or I need to make something clearer, or obviously if you can't hear me, make sure you do that too. I don't think this mic is on, so let me know if you have trouble hearing me. All right, so let's move on. We're going to jump into just a small part of chapter 6 and come back to the rest of the chapter later. We're going to talk about the part about how we grow bacteria in the lab. All right, so it's important in order to satisfy Koch's postulates that we are good at this. And it's taken a lot of trial and error in order to be good at it. And there's still a lot of bacteria that we aren't able to grow in the lab. So just some kind of basics of how we do that, a little terminology that we use. For instance, what's a pure culture? And then we're going to talk about some different kinds of media. So the media is what actually contains the nutrients that the bacteria use to grow. All right, so just some terminology that's we gotta, sometimes it takes some practice to use these words correctly, but this term culture, we can use that as both a verb and a noun. So if we're gonna say, I want to culture some bacteria, that just means we want to grow up that bacteria. All right, we want those few cells that we start to make many, many more cells. We want them to reproduce. If we refer to this as, oh, here's a culture, here's a bacteria culture, that just means the place where the microbes are growing. Okay, so that could refer to a test tube with media in it. It could refer to a petri dish with media in it. So here's a culture right here. Lots of bacteria growing. If we inoculate a culture, that means we're going to introduce an organism into a culture, or into the media. You'll be doing a lot of that in lab. And then the inoculum, okay, we use that as a noun, that's the actual microbe that's used to start the culture. Okay, so maybe you're using E. coli, for instance, to start the culture. That's your inoculum. And then the medium, which is the red in this picture here, that's just the collection of nutrients. So we're going to talk next about, or shortly, about what kind of things are in that medium that are going to enable us to grow up our microbes. Okay. And actually the plural there is media. Okay. All right, so a few other terms that we are using all the time. So once we inoculate a culture, typically we need to incubate it. All right, that means we're gonna keep it at some ideal growth temperature. For microbes that live in our body, we want to incubate them at body temperature. So maybe somewhere between 35 and 37 degrees Celsius, more commonly. So all the organisms that we grow in lab, we most often put them at about 35 degrees Celsius. And they're going to grow a lot more quickly that way. We'll talk a little bit about, there's all sorts of different microbes that grow at lots of different temperatures. But again, for the run-of-the-mill ones that we're using, about 35, 37 degrees. Okay, so sometimes it's easy to mix up those terms, inoculate and incubate, right? So we inoculate the culture first, and then we incubate it in a nice warm place. So here's an example of somebody, that blue circle there, that's a loop. Okay, the ones that we use in lab are usually metal because we heat them up and reuse them. That's a disposable loop. So here's somebody is inoculating some solid media here. So they probably dipped that loop into some liquid culture and they're spreading some cells on that solid media. You can't really see anything there. That's because there's only a few cells You can't see the cells with your naked eye. But once those cells have a chance to grow up, they typically grow into colonies. Now, not always, but most of the time, bacteria will grow in these discrete packets called colonies. So each one of these colonies has millions and millions of cells. Millions and millions of cells. So we can see them. They're big enough to see them with the naked eye. All right, so two basic kinds of media, liquid media and solid media. Okay, so we've been using liquid media for a long time before we developed solid media. We often refer to liquid media, another name for them is broths. Okay, so a broth culture, that's liquid media. And how do we make up liquid media? Oftentimes we will purchase whatever kind of media that we want. So here's some nutrient broth. It comes as a powder. You measure out the correct amount. And then you put it in a flask and put a certain amount of water in there. Sometimes you might stir it up to dissolve it, heat it up until it's sterile, and then you pour it into whatever container you want, usually a test tube. Okay, so liquid medias are good for growing large amounts of bacteria. Okay, so the bacteria has a lot of space to move around. There's plenty of nutrients. And until the waste products start building up, they're usually going to grow really quickly. And in fact, in lab, usually we only need to incubate overnight, and then we have just a ton of bacteria to work with. So how can you tell if there are bacteria growing in a broth culture? Usually the broth culture is going to be pretty clear. You can pretty much see through it if there's nothing growing in it when it's a But then the more bacteria are growing, the cloudier and cloudier that culture will get. And sometimes we get a bunch of cells to kind of settle out and end up on the bottom. So if you want to see if there's some growth there, you usually want to take the tube and kind of finger tap it to kind of mix it up. And any kind of thing you see swirling around in there, that's bacteria, right? So that's one of the big goals in your lab this week, Wednesday and Thursday. Inoculate a culture and see if you can get something to grow there and see if you can actually see it. Okay, sometimes it's hard to tell for sure whether there's something growing in there with a liquid media. So that's an advantage of using solid media, because usually it's pretty easy to tell if there's anything growing on solid media. Okay, so we'll say easier to see growth. So that's an advantage. And so how do we make solid media? Again, we usually buy it in a powdered form, add the water, heat it up, and then we pour it into whatever container we want it to be in. So if we pour it in a Petri dish, we usually just call those plates, and we let it cool and harden. Sometimes we might pour that molten media into a test tube and then kind of sit it at an angle to cool off and harden, and then we might inoculate on the surface here. We've got a little surface area called a slant. Okay, what makes that media hard? What's in there that's not in a broth? Well, it contains something called agar. And agar is a solidifying agent. Okay, so any recipe from microbiological media that has agar in it, it's going to be a solid media. What is agar exactly? It's a polysaccharide. It's actually derived from a type of seaweed. It's found in the cell walls of that seaweed. Okay, and we like to use agar as a solidifying agent because bacteria cannot digest. Okay, so that is what we want. If the bacteria can metabolize the agar, then it's not going to work as a solidifying agent anymore. So we want something that's inert and that's going to keep the media solid. And Robert Koch discovered this, or well, somebody in his lab that used it in her jams and jellies brought it to the attention of Robert Koch, and he's started making solid media, and we've been using it ever since. We haven't found anything that's better than agar. Okay, so it's kind of like, so it's a polysaccharide. It's kind of like cellulose. Cellulose is another polysaccharide. That's the reason we don't eat grass, because we can't break those bonds of the cellulose polymer and digest it. Okay, so it's kind of like that. I don't know the structure. I'd have to look at it of agar, but it can't be digested. Where do we get it? I didn't post this picture, but it's comes from red algae, which is a kind of seaweed that we haven't been successful in farming it. It just washes on shore some parts of the world. Like this was a picture from Morocco, Spain, Korea. People go out and collect it after storms, and then they get money for that. There was an article in Science about 5 years ago that was worried that we were over collecting it and that we were going to have not enough agar to make our media, but that really hasn't turned out to be a huge problem. But that's where agar comes from. Okay. So just a little bit more terminology. Most of the time when we're working with organisms in the lab, we usually want to be working with one organism at a time. And we call that having a pure culture. Okay, so your whole culture, your whole plate or your test tube or whatever has one species, one kind of organism in the, we'll say in the plate or test tube. Oops, test tube. Okay, so that's a pure culture. A culture that's not pure, we would call that a mixed culture. So let me come back to this slide in just a second. Here's an example of a mixed culture. Okay, so if you were to take a sample, say from the bottom of your shoe, take a swab and then streak it on some media, you might get something like this to grow. You would probably get all sorts of things. Looking at the different shapes and colors and sizes of the colonies there, there's maybe at least 12 different organisms on that plate. Okay, a mixed culture. If you were trying to identify any one of those, you would want to get it all by itself. Okay, so you would want to maybe take a little bit of bacterium 5 here, take another plate, streak it out on another plate, and get that organism all by itself before you are trying to identify it or characterize it in any way. Okay, so usually we want a pure culture, just one organism at a time. And we usually maintain that, or how do we get a pure culture? There's 3 different ways that we might streak out a plate. Okay, you might do a streak plate, a pour plate, or a spread plate. And I mentioned all 3 of those because you're going to do all 3 of them in lab, if you're in lab. Especially, you're going to do lots of streak plates. So that's a particular pattern that we streak out in order to just get those colonies all by themselves. Okay, isolated colonies. So here, each of these colonies, they're not touching any other colony. You got lots of isolated colony because this is an example of the streak plate here. So you've streaked out in a particular pattern in order to try to get those colonies all by themselves. And then you can look at those colonies and say, oh, they all look the same. There's most likely only one kind of bacteria because all those isolated colonies look the same. All right, so you'll be talking much more about that in lab. I'm not going to worry about these two because, again, I'll let your lab professor tell you about that. Once you have organisms growing on a plate, you want to not have anything contaminating it. So you're going to keep that pure by using aseptic technique, which you'll talk much more about in lab. So we're out of time. Thanks for coming and listening, and we'll pick up there on Wednesday. So have a good day.